Hello and welcome to the Beyond Biotech podcast, and this is number 6. I'm Jim Cornell from Labiotech.eu, and it's good to be back at work, although the heat wave, not so much. Fortunately, here in Western Scotland it's been warm but not overly hot, but anyone in an area that's suffering from the heat, you do have my sympathy. It's been a pretty busy week getting back into the swing of things after a few days off, although of course you can't just walk away from news, so I have been working intermittently, including last week's podcast. But this week was a lot of podcast interviews and meetings, and well, you know how it is when you get back after time off, you have twice the workload. The day trips with the family have been great, revisiting local waterfalls, mountains and beaches, and no disasters, so even better given how accident prone I tend to be. This podcast is going out on July the 22nd, which I am assured is National Pajama Day in the US, which may seem odd, but it's all for a good cause because it's a fundraiser to raise money for children living in foster care. And with more than $400,000 already raised, that can't be a bad thing. I think I'd raise more money from people telling me not to wear pajamas and wear my actual day clothes instead. I should let you know at this point who our guests are on the podcast this week, and it's a 3-interview, 4-person episode. We have conversations with Jeremy Skillington from Poolbeg Pharma, Louise Modis from Mogrify, and 2 people at ND Capital, Dani Bach and Eric Mosinger. And that brings us to some of this week's news you may have missed over at Biotech.eu, and there is quite a lot of news this week. Zyversa and Larkspur have merged to become a publicly traded biopharma company. Vice Bio has exited Stealth to work on a next-generation RSV vaccine, and we had an interview on investment opportunities with Sofinnova. We had an article on funding for a trial on lung immunity in elderly patients. Almirall put out a call for therapies for skin diseases, and Eiger received European approval for its progeria treatment. IVI and Moderna signed a deal on vaccine R&D. Big Hat Biosciences raised $75 million to design safer, more effective antibodies, and we had a story on plant-based nylon. LabShares Newton in the US has doubled its space for biotech startups, and it also launched a pitch contest. We looked at the top 5 organizations using Web3 in biotech. Alpenglow Bioscience and Core Plus partnered on taking 3D spatial biology to clinical R&D, and Cells for Cells treated its first patient with exosome-produced therapy for osteoarthritis. We had an interesting story on Spider Silk, a look at the biggest Asia-Pacific biotech investments in June, and Sinovac has started an influenza vaccine trial in Chile. 4051 Ventures closed a $43 million biotech fund and announced the first recipient. Oxford Science Enterprises has raised £250 million. We had some updates to a few older articles. There were a few sponsored articles as well, and you can read all of them and lots more at Labiotech.eu. Other things you can do at Labiotech.eu are listen to and subscribe to the podcast, watch videos, subscribe to our 3 times a week newsletter, and check out the editorial calendar which was recently posted, as we love to get contributions to the site, press releases, and suggestions for guests for the podcast. And so that brings us quite nicely to our first guest of the week, and the subject is hearing loss, something I know a little about personally, thanks to too many loud concerts, some of which I was playing in. And yes, I did wear hearing protection, but it does only go so far. And I'm not alone. According to the WHO, 430 million people have some kind of hearing loss, and that will be 700 million by 2050. So clearly it's a huge global issue. Mogrify has partnered with the Japanese company Astellas to address hearing loss, and it's a fascinating story about regenerative medicine, and rather than me try to explain it and fail, I will leave it to Mogrify's Chief Scientific Officer, Louise Modis, to tell us about it, And of course, I started out by asking for some information on Mogrify.
Yeah, great. Happy to do that. Thanks, Jim. So Mogrify has developed a suite of proprietary bioinformatic platforms to systematically identify transcriptomic and epigenetic switches that control cell fate and drive cell reprogramming. And this has been established based on a 12-year research collaboration between academics, and now built on most recently at Mogrify. What is really unique about Mogrify is that we can convert one cell into another. We can transdifferentiate any one human cell state into another cell state based on the rules that we can define with Mogrify. And our aim in the company is to use this knowledge and this power to drive in vivo reprogramming, endogenous reprogramming of cells that are missing and associated with specific pathologies and diseases. And of course, it can be used in many other applications as well, but this is where we aim to move forward with the company.
All right. Almost sounds like heading into the realm of science fiction in a way, doesn't it, to be able to reprogram cells?
The thing is that the need has always been there to reprogram cells. That need has always been there, and we just haven't, as scientists or medics, been able to do anything about it. We need, number one, to know what are actually the rules controlling cell fate and controlling cell state. If we know the rules for how to switch a cell from one state to another, we can apply them, because the applications from a practical point of view are evolving nicely. So the delivery applications and the different technologies for delivery are evolving. The delivery of gene therapy is evolving. The regulators and the clinical trials are evolving in lockstep with those gene therapy applications. So cell reprogramming is the next step if we knew what the rules were. And we as scientists have operated on the principle of biological knowledge or serendipity to find out a certain number of transcription factors or regulators that may control cell fate. And those have been advanced, and people are aiming to advance those in the clinic. But it is very heavily dependent on serendipity and our understanding of a particular biology. Being able to do it prospectively and say, I want to make a certain cell type. To do that, you need a systematic approach to actually look at all of the data, take the bias, the biological bias of our knowledge of biology out of it, at least in the first step, and then use that systematic knowledge to choose the very best combinations of factors that could be used to switch cell state. And that's where Mogrify comes in, and that's what we find really exciting about it, and allows us to switch cell state without going through developmental steps. So we don't have to take the cell through the many different steps that normally happen during development, but we can rather switch it to the cell state that you need in the context of disease.
And without giving away any trade secrets, I wonder if you could give me a little bit of a background as to how the cell reprogramming platform works.
Absolutely. So it is based on integration of public data sets in the first instance to give you a baseline of what genes are expressed in different cell types, and that's based on the Phantom5 data sets. The next layer is to integrate the regulatory networks like String and Mara that are used to interrogate protein interactions, protein and regulatory interactions between different systems to integrate that information. And then the next layer is to use RNA-seq data from, or ChIP-seq data from, or other sets of data from very specifically isolated cell types. So it's critical to know what you want to make in terms of the destination cell. It's critical to know what your starting cell is, what cells you want to convert. And it's critical to have a really pure population where you don't need many cells, but you need those cells to be pure, where you have RNA-seq data. And that data is then integrated into the system as well. So as I say, they've spent over a decade working on the algorithm and integrating the data. So those steps come together, and they identify, the algorithm identifies the top transcription factors that, if we select transcription factors, that's what it works with. The top transcription factors that are expected to drive the vast majority of the regulated genome in the target cell or in the destination cell. And then what's additionally very nice about Mogrify is that it integrates that information such that it tells you the combinations of transcription factors, the optimal combination to maximize the gene coverage. So you can choose a minimum set, for example, to minimize the number of things you introduce. You can also look at alternatives if transcription factors that are predicted are not interesting to be used for whatever reason, either from a safety point of view or from a patent point of view or from a conflicting biology point of view. We can use the algorithm to ask for alternative combinations to cover the same network of genes. And so it's really based on the transcriptional profile of the destination cell, not its ontogeny or its developmental path, but what it needs to look like in the end to be functional.
What kind of conditions and diseases is this applicable to that you're currently looking at?
Yeah, so we are looking at diseases where they are what we're calling first order diseases, where the disease or the condition is directly and completely associated with the loss of a particular mature cell type. So, for example, cone cells in the eye, hair cells in the ear, beta cells in the pancreas, in the context of the conditions where people have lost their vision, their hearing, or their ability to produce insulin, it's very clearly tied to that cell being missing. And we're targeting indications where they are postmitotic functional cells that don't normally regenerate in higher species, but where there is evidence of them regenerating in lower species. So one knows that the potential in principle is there to regenerate these cells. We just have lost the ability to do it from an evolution point of view. And we're also targeting diseases where the regeneration of those cells would be expected to last based on their postmitotic state. That if we can regenerate those cells in that function, that it would be expected to last. You wouldn't have to dose every month or every 3 months to have those cells. And that would allow us and will allow us to advance with AAV delivery in the short term. And as we evolve, we can tackle more complex diseases in the longer term.
You're working with Astellas. How did that collaboration come about?
So Astellas are investors in our company, and Astellas, of course, are a regenerative medicine company. They're really focused on evaluating and trying many different approaches to solve that problem in medicine. So they invested in us in our Series A, and therefore we were introduced through that to their scientific team. And we started conversations with them about what would be a collaboration that would be interesting for both of us to explore. And because Astellas has vast expertise in AAV delivery and vast expertise in clinical studies as well in the autology space, they complement us very nicely, where we can bring the molecular and cellular information around the transcription factors to drive the generation of hair cells and build on what's been done in the clinic already. So a single transcription factor has been taken into the clinic for hearing loss, and therefore both the practical steps in terms of delivering the factor and the regulatory steps in terms of working with the regulators to take that forward, there is precedent for that. But what we have observed is that, and I say we, the community has observed, is that one transcription factor was not enough to drive hair cell regeneration in humans. As we use our algorithm, we can see many different combinations to offer alternatives to a single transcription factor that we want to take forward and test with our colleagues at Astellas.
How will that develop in the future? I mean, what are the steps?
Yeah, so we are doing the bioinformatic step in the first instance, and generation of reagents and testing that those reagents work from a cell biology point of view. And Astellas brings their complex ex vivo model experience together with their clinical and regulatory experience. So there is a route to the clinic, and between us we have all of the components, and we will move it at pace to test the best combination in humans in due course.
And what's the end goal of this? Is it to cure hearing loss or to improve on what hearing old people like me still have left?
Yeah, so it is to target sensory neuronal hearing loss, which occurs in all of us, myself included, Jim, at this stage, where you lose the hair cells over time with just the natural aging process or associated with, for example, chemotherapy treatments, or exposure to very loud noises in the context of explosions and things like that. It is recreation of the missing hair cells to bring back hearing rather than to just preserve hearing that we still have.
So that's good news for people like me that play the drums and have absolutely destroyed their hearing in bands over the years with too much noise.
Yeah, exactly.
If you need anybody to volunteer for trials, then let me know.
Yeah, I think I'll be in line myself as well.
Are there any other collaborations that you're working on at the moment that you can tell me about anyway?
Yeah, absolutely. So the platform, as I've described earlier, the platform has the potential to figure out the rules that differentiate different cell states. And so it has actually really quite broad applications, from cell therapy, ex vivo, in vivo reprogramming, which we're focusing on internally, of course, and indeed other applications in terms of patient stratification and thinking about new target identification. So there are many applications for the platform. Our challenge is to remain focused on what we think is the most valuable application for Mogrify in the short to medium term, which is in vivo reprogramming of cells. So our intention is to focus heavily on that with a couple of projects internally. And in addition to the Astellas collaboration, we will, of course, keep our eyes out for any other collaboration that really fits squarely into the USP of the Mogrify reprogramming algorithm.
Next, thanks to technology that sometimes works, it's to the US and Switzerland at the same time for an interview with ND Capital. The company was in the news recently because of its second successful exit in the European biotech market when Galapagos acquired CellPoint and Abound Bio. To tell us about that, but first more about the company and its strategies, are two partners in the company, Dani Bach in Switzerland and in the US, Eric Mosinger, who we will hear from first.
So ND Capital is an early stage venture capital fund. We're a transatlantic group. We're based here in the Bay Area in Woodside, so right by Sand Hill Road, and also in Lausanne, Switzerland. And we cover both continents out of those two offices. We focus on what we think are important ideas across the life sciences and physical sciences, and I'm sure we'll drill down into a bit more detail, and focus on the early institutional rounds, seed, Series A. We're now investing out of our fourth fund. We closed it earlier this year. We've been around, I'd say, actively investing for 15 years or so.
And so for the expansion into Europe, when was that?
Yeah, so it's an interesting story. In a way, it's a re-expansion. So the firm originally started in Switzerland. The founder, Emmerich, is from Fribourg. He studied at the physical engineering at the EPFL, and then he launched Nanodimension out of Zurich. And then around 2008, we moved here to the Bay Area because we didn't see as many opportunities in Europe at that time. And so we invested predominantly in the US for about 10 years, and then re-expanded in Europe when we had the opportunity to partner with Patrick Ebischer and Dani, and we felt like the ecosystem had matured. And so we're excited to be back in Europe.
I guess the most recent news that came out of the company was about the acquisition of CellPoint. I wonder if you could give me a bit of history as to how that came about and what your involvement is in that.
So basically, it started when we met the founders, actually in a hike in Yosemite in National Park, around a JP Morgan conference. And at that time, it was just an idea that these guys had to bring the manufacturing of cell therapies at the point of care. They came with a prior experience in a cell therapy company called Gadeta that they transacted. And already at that time, they developed this idea that the big hurdle on cell therapy is manufacturing, and manufacturing centrally, and that doing it at the point of care would be much better, right? Faster, cheaper. So they came to us with this idea, and they had an agreement with Lonza to use the Cocoon machine that Lonza had developed. And this is basically a mini factory, automated mini factory that can do the whole cell therapy manufacturing process in less than a week, actually. And you can put these machines wherever you want, right? In the hospital. They had that, and they had an agreement with the hospital clinic in Barcelona for a license of a CD19 CAR T construct. These guys in the hospital, they had been trying in patients, in more than 100 patients actually, but manufacturing the CAR T cells by hand in their labs. So with the both elements and the idea, they came to us and they told us, look, would you find us? And so we did.
Could you tell me a little bit about the strategy that your company has when it comes to investing in biotech companies, like how you discover the companies or whether they come to you and what it is that helps you make the decision into investing?
So there are many sources, right? Many different sources. If I go into the companies that we have invested here, at least in Europe, Eric will comment on the US, many of them have come through personal networks. Here we are very committed into Series A, seed and company creation. And many of the companies that we have done have been through personal knowledge of the founders, of the professors in the academia that came with these projects. That's one, probably the most important source. And the other one has been proactively screening the literature, the patents, proactively going to the academic centers, the best academic centers in Europe, incubators, tech transfer officers, to see what is in there, right? What is cooking? What are those projects? And maybe the third source of deals have been through connections in the investor world, right? Other investor fellows and friends with whom we have worked in the past.
I think that's a great summary, Dani, and it's very similar here in the US. I think, yeah, a lot of great opportunities come through our network. But as Dani said, I think we've learned as a team that it's really important to be out there proactively looking for ideas. Otherwise, you risk kind of getting blinders, put on. So we have examples of investments we've made that started based on a publication we read and kind of reaching out to the PI, even though we didn't really know that person, and ultimately led to an investment.
Do the strategies between the US and Europe differ at all?
I think they do slightly, right? Because the markets are different. The way I see it is that here in Europe, although you have very good science, sometimes there are two things missing that may be more present in the United States. One, I would say, is the entrepreneurial spirit of some academics and scientists. Very often, you have to convince them to create a company, to file a patent, to get out of the academia, at least the project. And the second thing is experienced management teams that can run those companies. And in many occasions, for example, is individuals out of our group or out of our firm, ND Capital, that run those projects as project manager / interim CEO, at least at the beginning, until the project matures. We are able to attract more capital, but also more talent to those companies.
Yeah, I think we have a very similar strategy. We work together as one team across two offices, make decisions together. And it's actually turned into a bit of an opportunity for us, the situation that Dani described in Europe, where we've been able to do some company creation work, whereas in the US, you'll oftentimes have kind of fully formed teams to back.
You mentioned the company creation. Is that something that you have the ability to work with companies at very different stages of their journey, I guess?
We do, yeah, absolutely. And maybe I'll hand it over to Dani to talk about the earliest stage, the company creation part, then we can build from there.
Yeah. If your question is whether we have the opportunity to work with companies at different stages, yes, definitively. And it goes from really early stage at discussing with the professor in the university about what is the best way to run the experiments and to file the patents, and then helping them to start up the company, bringing the right management team. In some occasions, we are the only investors in these early phases. But then, of course, after that initial seed phase, further down the road, we keep supporting the company along all the life of the company until an exit, right, for us. It can come relatively quickly in some occasions, but in other occasions, it may need several rounds of investment and going even through IPO, and this is something we do, yes.
Is it just financial that you help with, or do you help them find board members and that kind of thing?
Excellent question. No, actually, the financial part is the less important, I believe, of everything that we can bring to the table, right? Because it's the less differentiated. Everybody can bring money to the table. Certainly, bringing everything a company needs to be built, this is what we try to bring. This includes talent, networks, people we know that can manage the companies, service providers that we know can help the companies. But also when time comes to exit those companies, to transact, putting the companies in contact with the right decision makers on the buyer side, for example.
The only thing I would add there is we have a number of kind of experienced management teams in our portfolio, and oftentimes there it's more about staying out of the way and helping periodically in a very strategic way, say around a financing effort or an IPO roadshow, or as Dani mentioned, a strategic outreach effort. But then we also have a number of first-time entrepreneurs in our portfolio companies, and they're going through this for the first time, and we can help guide and help them look around corners and hopefully avoid mistakes in many different ways. And of course, building a company, it sounds great, but it's oftentimes really hard. So also just providing moral support is an important thing along the way.
I guess we're talking about sort of the inception of companies and helping them get to the next step. What about things like your exit strategies? Because how do you decide when it's time to exit?
That's a very good question. Companies are not sold, they are bought, right? So ultimately it's the buyer who decides when they want to buy. As you know, there are two main exit routes, right? M&A and IPO. And these may be very, very different. The way you would build up a company to prefer one route or another may be very different, which, by the way, it could underline also the differences between European and US markets. Public European markets are certainly less rich and active and dynamic than the US public markets. So for European companies, if you go IPO, ideally should be NASDAQ, right? But then this increases the hurdle for a European company, right? Typically, you would like to build up American management teams, or at least management teams that are familiarized with American markets, and this is not so obvious, seen from this side of the Atlantic. And if you go IPO, if the idea is going IPO, you need to build up a more solid and broader company more often than if the idea is to go M&A, right? In which probably very focused asset companies, or at least companies that are very focused on a certain technology and a certain platform that can be appealing to the acquirer, would be rather the norm if you go M&A. So different exit routes call for different strategies in company building. Mm-hmm. Of course, you don't choose, right? So you have to be flexible, and at some point you may change the way you build up your company. You can start very lean, very focused towards an M&A, and ultimately, further down the road, as the company grows up, you may decide, okay, now it's time to think on an IPO. So let's broaden the pipeline, let's build up the team, and let's prepare for that route, which, by the way, is always interesting to keep optionality, right? And increase the competitive tension between both exit routes.
And of course, I think the IPO window isn't always open. So I think it would be difficult to build a company kind of banking on an IPO at a certain point in time. So as Dani said in the beginning, we focus on finding companies that we think can make a big difference and creating value. And then if you do that well, you hopefully will have the opportunity to choose between those two paths. And, I think it's worth noting that an IPO isn't really a true exit for us. Yes, our shares are publicly traded, but we look at it really more as a financing event for the company, and so that they can raise more capital. But then it does take a while to exit public positions. So it's a little bit of a different thing.
Is there anything else that you wanted to cover about the company that we didn't go into?
We are not only life sciences, we are also a physical science investment. Now you are talking to both of us who work on life sciences, but we have a very active team investing in physical sciences as well. Interestingly enough, as Eric said before, we're one single team deciding on all investments, physical and life sciences. And probably because of that, we get very interested as well in the convergence between these two worlds. At least from my experience in prior firms, it's pretty exceptional. Normally, you see two different teams deciding on one sector, maybe life sciences, and other sector, energy or tech or whatever. Here, we try to keep it together. Some of our best investments have been in the convergence of these two areas. More often than not is physical science innovation, engineering innovation applied to the healthcare market. It has worked very well for us with Twist Biosciences, for example, and NASDAQ listed. Squeeze Bio would be another good example of that. CellPoint, the company that we recently sold to Galapagos, another example of that.
And now from Switzerland and the US, we're headed to Ireland, to Poolbeg Pharma, a company that's been in the news at Labiotech.eu twice recently, First for a partnership with 13 Biotech on artificial intelligence, and earlier this week for approval for a trial on a treatment for severe influenza. To tell us all about the news and the company is Poolbeg Pharma's CEO, Jeremy Skillington.
So yes, a little bit about Poolbeg Pharma. We are an infectious disease focused clinical stage company. We're 1 year in existence. We spun out of Open Orphan, who are a clinical trials focused company. They run human viral challenge studies. And just about a year ago, we spun out. We raised 25 million sterling on the UK London AIM market. And we've been pushing forward both internal programs. Our lead program, POLB001, is a treatment for severe influenza, pushing that forward. About to start a phase 1B challenge study on that. We've also in-licensed other programs for respiratory viral infections, bacterial infections. And quite interestingly, we've got a whole wealth of human challenge viral data that we're analyzing using artificial intelligence through collaborations. So we've had a very active, very busy, very productive 1 year, and looking forward to many more years to come.
All right. And I noticed on your website, it mentions the fact that you can develop multiple products and also develop them faster and more cost effectively. Could you explain a bit about that?
Yeah, we believe so. And I think it's something that's unique, let's say, to the infectious disease space. And again, with our partners at Open Orphan, they've been doing this for years, where you can very quickly and very affordably run human challenge studies. You can get human data, which we believe, as a company from Poolbeg, our ultimate goal is to partner our programs early with pharma, and then pharma take them forward deeper into clinical development and ultimately onto the market. So we feel we can get human data very quickly. And a good example is the LPS challenge study we're about to begin. This is essentially 1 week dosing. So we'll have human data from a challenge study with 1 week dosing. And as you can imagine, with infectious diseases, some of them last quite long, but some of them are actually quite short, especially respiratory viral infections, where you get, whether it is influenza or RSV, you're quite sick for maybe a week or two and then you recover. So it's not like your traditional, say, biotechs that look at rheumatoid arthritis or cancer, which are longer treatments, longer clinical trials. So we have shorter clinical trials generating that early human proof of concept and then partnering with pharma. So our goal, our ultimate ambition, is to build a pipeline of programs that we bring forward, de-risk early. We'll invest, as I say, in these early clinical trials, relatively short clinical trials, and then partner with pharma. So partnering is our revenue generation method, where we'll have multiple products moving forward and then partnering regularly with pharma. So we'll ultimately get upfront payments and downstream milestones, royalties that pharma develop. So we'll do the de-risking, and then pharma takes on the heavy spend when it comes to later clinical trials, but we'll ultimately benefit. So we're going to have a conveyor belt of assets that we're moving forward. And again, in the infectious disease space is our primary focus. So again, these can be run relatively efficiently, effectively, quickly, and affordably.
So what do you currently have in terms of partnerships and pipelines? What the ones that you're allowed to tell me about, at least?
Yeah, our lead program, which we started with, let's say this is POLB001. This is a small molecule immunomodulator, and it's based on very exciting data from our colleagues at hVivo Open Orphan that observed that a target, an immune target called P38, was significantly upregulated in patients who had severe influenza versus those that had mild to moderate. So the ultimate goal is to develop a P38 inhibitor to demonstrate this early proof of concept in humans. And we're about to start a challenge study on that, and I'll speak a little bit about that. But it's essentially, this is a molecule we in-licensed from a Spanish group called Palau Pharma, bringing it forward into the clinic. It tackles the cytokine storm, the kind of overactive immune response to the presence of the virus. That's really the severe influenza condition. And but we feel that having generated data in humans, we can actually go back and apply this proof of concept to other diseases, other inflammatory diseases, where we know that P38 is a key driver. So as I said, that's our lead program, POLB001, which we've brought forward. We did state at IPO that we plan to initiate a clinical trial this summer. So again, happy to report that that's on track. So we're a team that, let's say, can execute on our commitments and execute on our promises. So our second product then, POLB002, in the respiratory viral space as well. But this is a broadly applicable prophylactic and therapeutic against a broader range of respiratory viral infections. This is a program we in-licensed from the University of Warwick, and it comes to us with a very robust preclinical package. So now we hope to move that forward again quickly into clinical testing. We've got some manufacturing to do and some regulatory discussions, and again to get that early human proof of concept. So this can have direct effect on influenza viruses as well. It essentially is a break on the replication of influenza viruses. So if there's an influenza virus presence, we have this RNA-based immunotherapeutic that kind of blocks the replication and blocks the disease in its path. But it also has benefits for treating other infectious diseases, such as respiratory syncytial virus, where it essentially triggers the nasal immune cells to be ready for an infection. So it kind of creates this innate immune response. So kind of, if you do happen then to get an infection, it helps block the disease from progressing. So very excited about that program, as they're moving that forward. And then a third program we have, POLB 003, is a vaccine that we've collaborating with the University College Dublin, and it's a vaccine against a gram-negative bacteria called melioidosis, or the disease is melioidosis. It's a pretty robust bacteria where it's quite resistant to antibiotics. It affects people mainly in the tropical, subtropical regions. And if you're unfortunate enough to catch this bacteria, and roughly 170,000 people do annually, there's a 54% fatality rate. So it's pretty striking, pretty dangerous bacteria. So we have quite an interesting, again, late preclinical vaccine against that particular disease. And again, importantly and excitingly, with our collaboration with University College Dublin, there's 5 other bacterial vaccines they're working on. So we have an option to those. So again, building our pipeline, and we're doing some diligence on those. They're a little earlier, but we see the potential to add those to our pipeline at the appropriate time. So that's more on the product side. And then on the platform side, which we again intend to kind of feed our pipeline, feed more products into our pipeline, we have an oral vaccine delivery platform, which again, quite interestingly, I think, we've learned a lot, through the COVID-19 pandemic of, how to treat, how to develop better vaccines, how the immune system responds to vaccines. The majority of people got injections in their arm that trigger systemic immunity, which is good. But I think there's an appreciation and an understanding that if you can trigger mucosal immunity, you'll stop the infection at the site of infection. So we want to trigger this mucosal immunity. So delivering these vaccines orally, which is a challenge. Your stomach acids and enzymes and proteases are very quick to degrade anything that's put in there. So we have an interesting technology collaborating with a group called Biotechnology, where they're able to protect the cargo and get it through the stomach acids and release it in the gut, and again triggering mucosal immunity in the gut, so you're blocking at the site of infection. So very kind of exciting. And again, as I say, this is a platform. This is not one product, so we kind of plan to kind of build a few assets, a few products coming from that platform. So very excited about that. So taking a step back, as part of our IPO, our spin out from Open Orphan, we have a license, we have access to their human viral challenge data. So a typical clinical trial that they run, they might have 60 volunteers that come in. And again, they could be in a setting where they either had a vaccine or getting a therapeutic. But we've got access to the data where these healthy volunteers, they get infected, they become sick, and then they recover. So we've got a whole host of data when it comes to gene expression and protein expression, signs and symptoms, blood samples, etc., nasal swabs. We have a lot of data that was ready to be interrogated. And again, difficult to do in a traditional or standard lab setting because you get so overwhelmed with the data. So we've signed 2 deals with 2 companies, one Cytoreason, who are based in Israel, around the influenza data that we have. And Cytoreason are a fantastic company to work with, really good collaborators, and very happy to work with little old Poolbeg, because our data is so, as they describe it, clean, because you have somebody who's healthy, you have their sample, somebody who becomes sick, you have their sample, somebody who recovers, and you have their sample. So you're able to track that immune system all the way through and how people are responding to the presence of viruses, what genes are up or down regulated, proteins appearing, disappearing. So you're able to interrogate that as, again, ultimately come up with new targets for diseases and new therapeutics to tackle these diseases. So very excited about that approach. And then our second AI collaborations with a US group, 13 Biotech, who are focusing on our RSV data, the respiratory syncytial virus data. And again, similarly, take this data set, very unique data set, and interrogate it using their algorithms. And they've completed stage one of collating all of the documentation now, or all the data, and now ready to interrogate. So we expect by the end of the year to have identified new targets and ultimately new drugs, or essentially reposition drugs that we can put into these treating these diseases. So again, this is very much cutting edge. I think the data I described, nobody else has this type of data. So it's quite unique for us to be able to interrogate this with our collaborators. So again, moving along well, regular phone calls, tracking progress, getting our scientific team involved. And as I said, it's going to be some very exciting new drug targets and drug candidates available to Poolbeg by the end of the year.
I guess the most recent announcement before the one this week is the working with 13 Biotech. Could you give me a little bit of background on that?
Yeah, I mean, it's a very interesting, when I joined the company just over a year ago, and they were telling me about, the data set that we had. And we did speak to maybe 8 or 10 different AI companies, who has the best capabilities, the best, algorithms to interrogate this data. And we talked to 13 about our approach, about their approach, and really, fantastic people to work with. So we begun, here's our data set. They were intrigued. And then you begin the negotiation. How we structure this? How will this work? And again, very good discussions all the way along. So we came to an agreement that, they would get access to all of our data. They would interrogate it. Anything that comes out of this that we develop, there'll be some downstreams, milestones and royalties to 13 Biotech. But ultimately, Poolbeg will be partnering these drugs with pharma who will take it forward for us. Again, our business model is quite unique. It's quite like Biotech in Germany, where they do the early research and development and then ultimately partner as the partner takes on the risk and the cost of development. But the 13 terrific, collaboration thus far, moving along well, happy with progress.
And I guess you have some news this week.
Yeah, we just got approval to begin, a regulatory approval to begin our phase 1B LPS challenge study. And we have indicated to the markets that, our plan initially, as I mentioned at IPO, was to have this trial begin in the summer of 2022. And yes, here we are. And again, happy we're meeting our goals, happy we're executing on our strategy. So this is an important study for Poolbeg. As I say, we've POB001, we want to demonstrate or generate proof of concept data in humans. And this LPS challenge study achieves this. And briefly, what this means is, again, taking in healthy volunteers, we give them a bacterial fragment called LPS, lipopolysaccharide, which the immune system recognizes as being foreign. So it thinks that there's an infection occurring. So it ramps up the immune response to tackle or target this foreign invader. This essentially mimics what happens if you get a viral infection such as influenza. So your immune system is ramping up and triggering all kinds of gene expression and protein expression. So LPS, let's say, is a mimic of the similar response. We'll begin recruiting healthy volunteers to essentially give them LPS to trigger the immune response, but also give them POB001 to block or to blunt that inflammatory response. So again, this gives us data, and we will have this data before the end of the year that showing that in this inflammatory response, in this infectious disease equivalent setting, that POB001 will block this response. And again, there'll be that corollary that this can also be successful with infectious diseases such as severe influenza. So again, severe influenza is a direction we're taking because of data generated, and we filed intellectual property that PTR inhibitors can have a significant effect or an impact on severe influenza, and that these patents go out to 2038. So we're well protected in the patent space. So again, generating this data, which we'll have by the end of the year, I think will be very attractive from a partnering standpoint. And again, that's the ultimate goal, where we'll be partnering with pharma to take these programs forward. I think that this data set at the end of the year should assist us then in partnering in 2023 discussions with pharma. They're aware of what we're doing, so we'll kind of go dive deeper into the data and again, as I say, ultimately partner with pharma in 2023 for POB001.
And that does it for another podcast. Some really cutting edge information and some great guests this week. I'm sure you will agree. Next week is still a little bit up in the air. I do have some interviews done already, but we may push them back a week because my colleague, Liza Laws has done a couple of interviews for the podcast that relate to hepatitis. And next week, starting July 26th, is Hepatitis Awareness Week. And World Hepatitis Day is the 28th. So it's quite possible we may end up with a special podcast related to that. It's a nice issue to have, though, with so many interviews, and there's such a lot going on globally in biotech. Here at home, the warm weather ended today, and we have a forecast of 4 days of rain, so that's really going to put a wrench in any walking plans this weekend. Or I guess we could just get wet. Sadly, the rain here very rarely comes down perpendicular to the ground thanks to so much wind, and the ground also gets very soggy very quickly. When we first moved to Scotland a few years ago, we were very impressed by how hardy the people seemed to be, because they didn't use umbrellas. Well, 2 days and 3 snapped umbrellas in the wind later, we found out why. Anyway, we'll try and enjoy the weekend regardless, and I hope wherever in the world you are, you do the same, and that you'll join us again next week for another Beyond Biotech.